Welcome to the first video in the series covering the final stage of the eukaryotic cellular respiration process, which is what we call the electron transport chain, which we will henceforth abbreviate as the ETC in order to save time. Now, to orient ourselves about where the electron transport chain takes place, you can see that there is a phospholipid bilayer here with several large multi-protein complexes all embedded within this membrane. Now, this is not actually the cell membrane, but this is actually the inner membrane of the mitochondrion, which means geographically the mitochondrial matrix is here, and this is where Krebs cycle is taking place. Now, this geographic orientation is important to note based on what the electron transport chain actually does, because all of that NADH that we produced in both glycolysis, if you'll recall from glycolysis, the example of NADH production is the dehydrogenation of G3P, but the majority of all NADH produced in cellular respiration is done in Krebs cycle in the very mitochondrial matrix that is immediately adjacent to the inner membrane. And what we want to use this NADH to do is to produce ATP using this very electron transport chain here. So how exactly does this work? So each of these membrane complexes, because it isn't a single protein, each of these is referred to by a simple name based on where they occur in the electron transport chain. So this first complex here is simply what we call complex 1. And then the complex that comes after it, not surprisingly, is called complex 2, then we have complex 3 and complex 4, not exactly brain surgery we're doing here with this naming convention. What is less immediately obvious is what the names of each of these complexes are, because that actually gives us a hint about what they do. So the proper name of the complex 1 group of proteins is what we call NADH dehydrogenase, meaning that it is going to react with NADH, both that exist from glycolysis, but primarily the NADH that is produced in the Krebs cycle is going to enter the active site of this multi-protein complex and release its H+. And again, given that this enzyme is called dehydrogenase, that shouldn't exactly come as a surprise. Now, when the H+ is released, the 2 electrons that NADH is carrying are also going to be released, and the 2 electrons are actually taken by a number of the proteins that function individually as electron carriers. And the reason why we call this the electron transport chain is that all of the pairs of electrons that are released by every NADH molecule are transferred from one protein in the chain to the next, to the next, to the next, and so on, because each protein within the chain has a greater attraction for electrons than the previous protein did before it. What that means is that if we measure the attraction to electrons, which is what we know as electronegativity, each protein whose job it is to carry electrons through the electron transport chain has a greater electronegativity and therefore a greater ability to attract electrons than the one that came before. And as these electrons move, the movement of these electrons creates a relatively strong electrical kinetic energy, which we can then use a number of these proteins in order to accomplish a specific goal. Now, complex 1, complex 3, and complex 4 are all examples of H+ active transport proteins. So when we normally think of active transport, we think of proteins that require energy in order to transport substances from a low concentration into an area of high concentration using an energy source, usually ATP. But remember that the goal of this whole process is to make ATP, so it probably doesn't make sense that each of these active transport proteins uses ATP as a power source. Instead, the kinetic energy of the movement of the electrons released by NADH is what supplies the energy. In other words, each of these electron carrier proteins, as the electrons are transferred from protein to protein, these proteins are creating an electrical current that is literally used to power each of these transport proteins. And every time a pair of electrons flows through each of these transport proteins, these proteins can be used to grab the H plus that was released by NADH and pump it into the intermembrane space, which is the space in between the inner mitochondrial membrane, where the electron transport chain is occurring, and the outer mitochondrial membrane outside of the intermembrane space. Now, as a consequence of this, all of the H plus inside of the intermembrane space is going to become concentrated, because the more electrons that flow through the electron transport chain, the more these transport proteins are activated, the more H plus is pumped into the intermembrane space, and the higher the concentration becomes, therefore creating a hypertonic situation. Now, it's important to note that there is actually an outer membrane here, because this outer membrane prevents all of the H plus in the intermembrane space from being able to get out. And as a consequence of this, all of this H plus eventually wants to diffuse back into the mitochondrial matrix, because as the concentration of H plus decreases, these transport proteins are creating a hypotonic environment, which will naturally make the H plus outside want to diffuse back inside. But that's a subject for the following video. Now, one additional detail that we can talk about, we've talked about what the eventual target of NADH is, but we've never talked about what the purpose of FADH here is. So we can refer to this multi-protein complex as FADH dehydrogenase, although it goes by the term fumarate reductase, referring to a specific substrate in Krebs cycle from which FADH is produced. Simply put, NADH and FADH2 have different structures because of this, that means that FADH2 is not able to fit into the same active site that NADH enters because of their differences in structures, and therefore FADH2 cannot use the complex 1 proteins NADH dehydrogenase, so instead they release, or FADH2 releases its electrons to complex 2, which we find here. And as a consequence, the electrons from FADH2 are only able to stimulate complex 3's transport proteins and complex 4's transport proteins, and consequently NADH will result in more H plus being pumped into the intermembrane space than FADH2 is, simply because of the difference of where they release their H plus and electrons. Now, the one other question that we need to answer is, what is causing the electrons to get from the beginning of the electron transport chain to the end? Well, the answer has to do with electronegativity. And if you consider what the single most important element is in order to keep humans or any aerobic life breathing and prevent them from dying after 4 minutes, well, I kind of gave away the game that this is where oxygen gas is important. So by the time that the electrons from NADH and FADH2 make it to the end of complex 4, to the end of the electron transport chain, we need a very, very strong element in terms of its ability to attract electrons. And as it turns out, oxygen has the second highest electronegativity on the periodic table of between 3.4 and 3.5, depending on where you look. But the point is, only fluorine is better at gaining electrons than oxygen is. And as a consequence of this, because of oxygen's ability to gain electrons very easily, we refer to oxygen as the terminal or final electron acceptor that is essential for the electron flow in the electron transport chain, and we can actually illustrate what happens here. So when oxygen gains electrons, this results in the creation of oxygen in its ionized form. Now remember that all of this H plus that we've pumped into the intermembrane space isn't going to stay there. Eventually it's going to make its way back into the mitochondrial matrix, where it can react with the negatively charged oxygen ions in order to make water, and this is where the water that is produced at the end of cellular respiration comes from. Now in the next video, we're going to examine what the H plus is specifically used for. So in closing to this video here, we can talk about a little bit of the terminology that's used for the electron transport chain. So each electron transfer that occurs from one protein in the electron transport chain to the next, to the next, to the next is defined as what we call a redox reaction, which is actually a compound word describing two different types of electron transfer reactions. Now, if we look at the very beginning, NADH, when it reacts with complex 1, NADH is going to lose electrons, and therefore we say that NADH has become oxidized because it has lost electrons. So, loss of electrons, Le, is equal to oxidation, while complex 1 is the half of this reaction that gains electrons, and therefore we say that it has been reduced. And the easiest way to remember what reduced means is that if complex 1 is neutral, if it has a charge of 0, if it gains 2 electrons, now it has negative charge, so the overall charge has gotten more negative and therefore reduced. And there's a good way for you to remember the distinction between these two, where Leo, if we pretend that Leo is an imaginary lion, loss of electrons is equal to oxidation, and what noise do lions make? Well, a gain of electrons is equal to reduction. Lions go grr. If you thought that that pun was kind of lame, that's exactly the point, because now it is forever etched into your brain and you will never forget it. So the point of discussing this is when we say that the electron transport chain is driven by electronegativity, that's only partly true. A better way of explaining this is that each protein that occurs within the electron transport chain has what we call a higher reduction potential. And now that we know what the word reduction means, having a higher reduction potential means that each protein within the electron transport chain gains electrons more easily than the one that came before, and this is right in line with the electronegativity model. A different way of looking at this is looking at it in terms of energy, referring to each transfer of electrons from one protein to the next, to the next, to the next is more spontaneous, because each electron carrier protein in the chain wants the electrons more than the one that did before, because they are able to gain electrons more easily, hence the term reduction potential. Now that we've explored the literal electron transport chain in great detail, the next video will look at the fate of H+ in the intermembrane space and how that is used to create ATP.